Imagine a machine, and not a clean digital machine made of silicon and, perfect logic gates.
Hmm.
Imagine a machine built of biology, a wet, chaotic electrical storm that has something like 100 billion components.
It's a number that's hard to even conceptualize.
And the really terrifying part isn't the number itself. It's that these components, these neurons, they aren't just wires. They're not just passively carrying a signal from point A to point B.
No, not at all. Each one is a decision maker.
Yes, each one is like a little sovereign state, constantly voting, debating, rewriting its own rules in real time based on, I don't know, thousands of whispers and shouts it's getting every single millisecond.
It creates a scale of complexity that is, well, frankly humbling. You're talking about the central nervous system and specifically the challenge of systems pharmacology.
Right.
When we look at the heart, we can model it as a pump. The kidney, it's a filter. But the brain, it regulates everything from the simplest reflex, pulling your hand away from a hot stove, all the way up to the abstract construction of consciousness.
And yet, our job, or at least the job of the text we're covering today, is to figure out how to tune this engine with a single molecule. It feels a bit like trying to repair a supercomputer by just pouring a chemical soup over the motherboard and hoping for the best.
That's a very, very apt analogy because unlike a computer where all the connections are soldered and fixed, the connections in the brain are chemical. And that right there, that's our entry point.
Welcome to the deep dive. Today we are tackling neurotransmission in the central nervous system. We are synthesizing two giants of the field. Katzung and Trevor's Basic and Clinical Pharmacology, specifically their chapter on the pharmacology of CNS drugs, and Goodman and Gilman's The Pharmacological Basis of Therapeutics, Chapter 14.
And this is such a powerful combination. Katzung, it gives us that clinical scaffolding, the what and the where. But Goodman and Gilman, that gives us the molecular how. So our mission today isn't to just list a bunch of drugs. We want to help you build a mental model of systems pharmacology. We really need to understand how integration happens, what the difference is between fast and slow signaling, and maybe most importantly, how the brain adapts to our interventions.
We're going to build this thing from the ground up. We'll start with the cellular machinery, then move to the electrical language the cells use, and then dive really deep into the chemical signaling that, in a way, defines who we are.
And just a quick reality check for everyone listening.
Yes.
This is an interpretive synthesis. Think of it as a conceptual map to help you navigate the territory. But you still have to walk the path yourself. You absolutely need to read the primary texts to get all the granular detail.
Think of this as the architectural blueprints. You still have to lay the bricks.
Exactly.
Okay, so let's start with that foundation, the cellular stage. I think most people listening probably have a mental image of a neuron, the dendrites, the cell body, the axon. But Goodman and Gilman makes this great point that structurally, neurons are just incredibly diverse.
Oh, massive.
But functionally, they all face the same problem, which is integration.
Precisely. We shouldn't think of the neuron as a simple relay race runner just passing a baton. The neuron is a computer. The dendrites, that huge receiving tree, they collect thousands of inputs. Some are excitatory, they say fire. Some are inhibitory, they say stay silent.
And these signals all kind of wash over the cell body, the soma.
They do, they all converge on the soma. But the critical decision, the go or no-go vote, that happens at a very specific, very important location called the axon initial segment, the AIS.
Right, the trigger zone. So why is this specific spot so important, pharmacologically speaking?
Because the AIS has an incredibly high density of voltage-gated sodium channels, just packed with them. It has the lowest threshold for firing in the entire cell. So the soma acts as this integration center. It sums up all these little electrical potentials washing over the membrane.
And if that sum, that total vote, by the time it reaches the AIS, is enough to crack open those sodium channels?
You get an action potential, an all-or-nothing spike. If it's not enough, the signal just fizzles out right there.
So the neuron is basically filtering noise. It's ensuring that only a significant synchronized wave of information actually gets passed on.
That's the idea.
But the neuron isn't the only player on the stage.
Yeah.
Katzung is very clear that we have to talk about the supporting cast, the glia. And honestly, calling them a supporting cast feels like a huge disservice, especially since they outnumber the neurons.
It is a massive disservice. The old view was that glia were just glue, inert packing material holding the important bits in place. The modern pharmacological view is that you cannot have a functioning synapse without them.
Okay, so let's take the astrocytes, for example. I
Right.
used to think they just provided, what, structure and nutrients.
Right. But Goodman and Gilman really details their active role in what's called the glutamate-glutamine cycle.
This is such a key mechanism, so walk us through that, because it's not just about cleaning up a chemical spill, is it?
No, it's a vital metabolic handshake. It's a partnership. When a neuron releases glutamate, which is the brain's main excitatory go signal transmitter, it can't just hang around in the synapse.
Why not?
Because if it builds up, it causes excitotoxicity. It literally excites the next neuron to death. It's toxic in high concentrations. So the astrocyte uses these incredibly high affinity transporters to basically vacuum up all the excess glutamate.
Okay, so it cleans it up, but it doesn't just store it or break it down.
Correct. This is the elegant part. Inside the astrocyte, an enzyme called glutamine synthetase converts that dangerous glutamate into glutamine, which is basically inert. It's harmless.
So it neutralizes the threat.
It neutralizes it, and then the astrocyte ships this safe glutamine back to the neuron. The neuron then takes it, converts it back into glutamate, and repackages it into vesicles to be used again.
Wow. So it's a complete recycling system.
A perfect closed-loop recycling system. And if you disrupt this cycle, say if that enzyme fails during severe liver failure or in specific metabolic crises, the brain can enter a state of toxic hyperexcitability. The astrocyte is arguably just as important for that signal as the neuron itself.
That's a fantastic example of a system dependency. Okay, then we have the oligodendrocytes. These are the insulators.
They create the myelin sheath in the CNS. The physics here is pretty straightforward. Insulation increases the resistance across the cell membrane, which forces the electrical current to jump down the axon from node to node rather than just leaking out.
It's like putting plastic insulation on a copper wire.
Exactly. It increases the conduction velocity from a slow crawl to over 50 meters per second. And the clinical relevance here is just stark, think multiple sclerosis. In MS, the body's own immune system attacks these specific cells, the oligodendrocytes.
So the myelin gets stripped away.
It gets stripped away and the signal just leaks out. The transmission slows way down or stops entirely. And that's what leads to the sensory and motor deficits we see in patients with MS. It's a failure of this one specific cell type.
And the third type, the microglia. These are the immune cells, right? Derived from macrophages. But the texts mention something absolutely fascinating about them. This idea of synaptic pruning.
This is where immunology meets psychiatry. It's a really hot area of research. Microglia are mobile. They patrol the brain like little sentinels. They eat debris, dead cells. But during development and even in adulthood, they actively remove synapses that aren't being used efficiently.
So they're like a gardener, pruning a rose bush to make the remaining bloom stronger.
That is the perfect analogy. It's a use it or lose it system enforced by the microglia.
But the implication in the source material is that this gardener can sometimes go rogue.
Precisely. There is a growing body of evidence, which both texts highlight, linking microglial dysfunction to diseases like schizophrenia and Alzheimer's. The hypothesis is that in some of these conditions, the microglia might be over-pruning.
So they're eating healthy connections.
They might be effectively eating memories or disconnecting functional cognitive circuits. It completely reframes these diseases. They're not just a chemical imbalance anymore, but potentially a neuroinflammatory or structural problem.
That completely changes how we might even begin to look for future drugs. Okay, so before any drug can interact with these astrocytes or neurons, it has to get into the building in the first place. We have to talk about the blood-brain barrier, the BBB.
The BBB is the absolute bane of the CNS drug developer's existence. It's not just a wall, it's a hermetic seal. The endothelial cells that make up the capillaries in the brain are stitched together by these things called tight junctions, and then they're wrapped in those astrocyte end feet we just mentioned. There are no pores, no gaps.
It's a fortress. So if you're a drug molecule floating in the blood, how on earth do you get in?
You basically have two options. Option one, be hydrophobic. If you are liquid soluble, you can just dissolve right through the cell membranes. You can ghost right through the wall. This is how general anesthetics, alcohol, many sedatives, this is how they work.
And option two, if you're not lipid soluble.
You have to hijack a transporter. The classic example from Katzone is in the treatment of Parkinson's disease. In Parkinson's, you need to replace dopamine in the brain. But dopamine itself is a polar molecule. It can't cross that lipid barrier. It just bounces right off.
So we use a Trojan horse.
We use a Trojan horse named L-DOPA. L-DOPA is a precursor to dopamine, and it looks just enough like an amino acid that it tricks the large neutral amino acid transporter, or LAT1, into carrying it across the barrier.
Like it's sneaking in with a grocery delivery.
Exactly. And once it's safely inside the brain, an enzyme converts it into the active dopamine that we need. We are literally leveraging the brain's own nutrient delivery system to smuggle in our medication.
Is the barrier absolute, though? Are there any leaks in this system?
There are a few, and they're there for a very good reason. We call them circumventricular organs. The most notable one is the area postrema, which is located down in the brainstem. It lacks a tight BBB.
Why on earth would you leave a door unlocked there?
For security, the area postrema acts as a chemical sniffer. It's like a smoke detector for your blood. Its job is to constantly sample the blood for any circulating toxins. If it senses something dangerous, it immediately triggers the vomit reflex to expel the contents of the stomach.
Which explains why so many dopamine drugs like L-DOPA or apomorphine cause such severe nausea and vomiting.
Exactly. They are stimulating this exposed, unprotected nerve center before they even get a chance to help the rest of the brain. The side effect is born of the anatomy.
Let's move to the signal itself. We've set the stage, we've got the security system. Now, let's talk about the transmission. Section 2, the language of voltage. The currency of the brain is the flow of ions.
It is, and we need to be really precise here. The ions, sodium, potassium, calcium, chloride, they have what's called an electrochemical driving force. They want to move to equalize either their concentration or their charge. The neuron's job is to control this flow using voltage-gated channels.
Goodman and Gilman breaks these down beautifully. Let's look at the big three when it comes to the action potential. Sodium, calcium, and potassium. Sodium is the spark, right?
Sodium, or Na+1, is the absolute driver of depolarization. When that voltage threshold is reached at the axon initial segment, these voltage-gated sodium channels pop open, and sodium rushes in because it's highly concentrated outside the cell. This massive influx of positive charge is the spike of the action potential.
But these channels, they don't just stay open forever. They have a kind of a built-in timer.
They do. They enter what's called an inactivated state. It's like they close and then lock for a brief moment. And this is crucial for pharmacology because many of our best anti-epileptic drugs like phenytoin or carbamazepine, they specifically bind to these channels only when they are in that inactivated state.
So they don't block normal firing.
Exactly. They prolong that timeout, which prevents the neuron from firing too rapidly, like in a seizure, but they don't really affect normal lower frequency firing. It's a very clever state-dependent block.
That's a fascinating nuance. Okay, now calcium, K2 plus PSA. I always find calcium interesting because we usually talk about it as a signaling molecule inside the cell, but here it's also a charge carrier.
It's the bridge. It's the great translator. The action potential is an electrical signal moving down the wire. But the release of a neurotransmitter, that's a chemical event. Something has to translate the electricity into chemistry. That something is calcium.
How did that work?
When the electrical spike hits the nerve terminal, it opens these high-voltage-gated calcium channels, mostly N-type or PQ-type. Calcium rushes into the terminal, and that specific chemical signal interacts with the docking machinery, the snare proteins, to cause the vesicles to fuse and dump their transmitter into the synapse.
So no calcium.
No release. Zero. The signal dies right there at the terminal. End of the line.
And finally, potassium, K plus R. The brake.
Potassium is all about repolarization. It's the reset button. After the sodium rushes in, these potassium channels open up to let potassium flow out of the cell. Since potassium is a positive ion, losing it makes the inside of the cell negative again. It resets the membrane potential, getting it ready for the next signal.
And if you block these channels...
If you block potassium channels with a drug like 4-aminopyridine, for instance, you delay that reset, the action potential gets much wider, which means more calcium has time to enter, and you get a massive release of neurotransmitters. It's exciting, but often it's too exciting. It can lead to convulsions.
I just love the section in G&G about toxins. It feels like a detective story. I mean, we didn't just find these channels under a microscope. We sort of deduced their existence because nature kept inventing new ways to kill us with them.
It's a history of biological warfare, and we've learned so much from it. Take alpha-bungarotoxin. It comes from the venom of the many-banded krait, a snake. It binds with just incredible specificity and tenacity to the nicotinic acetylcholine receptor at the neuromuscular junction.
So tightly that it's basically irreversible.
Exactly. It binds so tightly that it allowed scientists back in the 1970s to physically fish these receptors out of tissue samples and purify them for the first time. We owe our entire molecular understanding of that receptor to that one snake.
And what about tetrodotoxin, TTX, from the pufferfish?
A completely different beast. TTX acts like a cork in a bottle. It just sits right in the outer mouth of the voltage-gated sodium channel and physically occludes the pore. Sodium cannot get in.
So no action potentials can generate.
None. And that's why fugu poisoning leads to this terrifying conscious paralysis. Your brain is working perfectly, but the wires going out to your muscles are completely dead.
Nature really is the best pharmacologist. Okay, so the signal travels down the axon, calcium comes in, the vesicles fuse, and the neurotransmitter is released into the synaptic cleft. Now we are at the decision point again, the postsynaptic potential.
Right. And here we need to distinguish between the signal itself, the neurotransmitter, and the result it produces. The result is either an EPSP, an excitatory postsynaptic potential, or an IPSP, an inhibitory postsynaptic potential.
And this depends entirely on which type of ion channel opens, correct?
100% correct. If the neurotransmitter binds to a receptor and opens a channel that lets positive ions like sodium or calcium in, the inside of the cell becomes less negative. It moves closer to the firing threshold. That's an EPSP.
The go signal.
The go signal. If, on the other hand, it opens a channel that lets positive ions, like potassium, out, or negative ions, like chloride, in, the cell becomes more negative. It moves further away from the threshold. That's an IPSP.
The stop signal.
The stop signal.
Katsung talks about summation here, which brings us back to that voting concept we started with.
Right, spatial and temporal summation. Spatial summation is like being at a town hall meeting where 50 different people standing in different parts of the room all yell yes at the exact same moment.
So the inputs are physically spread out across the dendrites, but they arrive at the same time.
Yes, and the soma just adds them all up. Temporal summation is different. That's like one person right next to the microphone shouting, yes, 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 incredibly fast. A single input firing at a high frequency can pump ions in faster than the cell's pumps can clear them out, ratcheting up the voltage step by step.
So the neuron is constantly doing this calculus. Do I have enough yes votes, either from many sources at once or one source very fast, to outweigh all the no votes?
Exactly. And a drug, a CNS drug, fundamentally tilts that election. A sedative like Valium doesn't shut the brain off. It just finds a way to stuff the ballot box with no votes by enhancing those IPSPs.
This is a perfect segue to the receivers themselves. Section 4, receiver types. The texts make a really hard distinction between ionotropic and metabotropic receptors. And this isn't just some minor structural detail. It really defines the speed of thought.
It defines the entire timescale of brain function. Ionotropic receptors are built for one thing, speed. They are ligand-gated ion channels. The key thing to remember is that the receptor is the channel.
It's a single protein complex.
Right. The neurotransmitter binds, the protein twists, and a gate in the middle of it opens. Boom, ions flow. We are talking on the order of milliseconds.
This has to be for things like reflexes, sensory processing, motor control, things that need to happen right now.
Absolutely. The nicotinic receptor, the GABA-A receptor, the glutamate AMPA receptor. This is what we call fast synaptic transmission. It's direct, it's quick, it's point-to-point.
But then we have the metabotropic receptors. These are the G-protein-coupled receptors, the GPCRs. And honestly, looking at the mechanism, it just seems absurdly complicated compared to the simple open-the-gate model.
It is complicated, but it's complicated for a reason. So let's walk through the mechanism. The neurotransmitter binds to the receptor on the outside of the cell. This causes the receptor to change its shape, which in turn activates a G protein that's waiting on the inside of the cell. That G protein then sheds a GDP molecule and picks up a GTP, which activates it. It splits apart. The alpha subunit goes off and finds an effector, like the enzyme adenyl cyclase. That enzyme starts churning out a second messenger, like cyclic AMP or SCAMP.
And we're still not done.
Not even close. That TMP then activates a protein kinase, like pKa, which then goes on to phosphorylate dozens of other proteins or ion channels, changing their function.
That sounds absolutely exhausting. Why, evolutionarily, would we ever use a system that takes seconds or even minutes to produce an effect?
Two crucial reasons: amplification and duration. With an ion channel, it's a one-to-one transaction. One neurotransmitter molecule opens one gate. That's it, with a GPCR.
The cascade.
It's a massive cascade. One neurotransmitter activates one receptor, which can activate, say, 10G proteins. Each of those can activate an enzyme that produces thousands of CAMP molecules, and those can activate kinases that affect tens of thousands of downstream targets. You get this enormous biological signal from a tiny initial input.
And what about the duration?
These changes last. They can even move all the way into the nucleus of the cell. That protein kinase, pKa, can phosphorylate a transcription factor called CREB, which then changes gene expression. It can literally rewrite the protein makeup of the neuron over hours or days.
This is the aha moment for pharmacology. This right here explains why a drug like Xanax, which acts on an inotropic GABA-A receptor, works in 20 minutes, but an antidepressant like Prozac, which acts through metaprotropic serotonin pathways, takes 4 to 6 weeks to have a therapeutic effect.
That is exactly it. Xanax is just opening a door. Prozac is ordering a full-scale renovation of the house. The antidepressant effect isn't from the initial binding. It depends on these long-term downstream changes boosting things like BDNF, brain-derived neurotrophic factor, growing new synapses. You are waiting for the biology to adapt, not just for the drug to bind.
That is such a crucial distinction for listeners to grasp. Okay, let's meet the cast of characters. Section 5, the neurotransmitters. The texts divide these into the heavy hitters, the amino acids, and the modulators, the monoamines. Let's spend some real time here, because this is the bread and butter of our drug targets.
Let's start with those heavy hitters, glutamate and GABA. Between these two, they handle the vast majority of all information transmission in the brain. They are the yin and yang of the CNS.
Glutamate is the gas pedal. It is the primary excitatory transmitter.
Almost every neuron that projects information from one brain region to another uses glutamate. It acts on several different receptors, but we really need to focus on the dynamic duo, AMPA and NMDA receptors.
AMPA is the workhorse, right? Just the basic fast signal.
Yes. AMPA receptors are inotropic. Glutamate binds, they open, sodium comes in, the cell depolarizes. They handle the basic faithful transmission of the signal from one cell to the next. But NMDA is special. Katsung calls it out specifically for its central role in plasticity.
And the NMDA receptor is often described as a coincidence detector. You have to explain the magnesium block because that is the key mechanic here.
Right. So under normal resting conditions, the pore of the NMDA receptor is physically plugged by a magnesium ion, Mg2 plus Seth. At the cell's resting voltage, that magnesium is stuck tight. So even if glutamate binds to the outside of the receptor, the channel won't open because the magnesium is in the way.
It's like having the right key for a lock, but the door is bolted shut from the other side.
A perfect description. So how do you unbolt it? You have to depolarize the membrane first. If those AMPA receptors fire enough and make the inside of the cell more positive, that positive charge inside the cell will physically repel the positively charged magnesium ion and kick it out of the pore. Now the channel is unblocked.
So NMDA receptors only activate when two things happen at once. The presynaptic neuron releases glutamate, and the postsynaptic neuron is already excited.
Exactly. It detects that presynaptic release coincided with postsynaptic depolarization. And when they finally do open, they let in calcium. And that influx of calcium is the trigger. It's the signal that tells the cell to strengthen that specific synapse.
How does it do that?
It sets off a cascade that inserts more AMPA receptors into the membrane, making the synapse more sensitive to future glutamate release. This is long-term potentiation, or LTP. This is widely believed to be the molecular basis of learning and memory.
The old saying, neurons that fire together wire together. The NMDA receptor is the welder that makes that happen.
Beautifully put. And clinically, this is where a drug like ketamine works. Ketamine actually enters the open NMDA pore and blocks it. It basically replaces the magnesium with a much stickier, longer-lasting plug. It disconnects you from your sensory input.
Okay, now for the flip side of glutamate, GABA, the universal brake.
GABA, or gamma-aminobutyric acid, is the main inhibitory transmitter in the brain. And just like with glutamate, we have two main receptor types to consider, GABA and GABA-B.
GABA is the famous one. It's the target for so many of our sedatives and anxiolytics.
Right. GABA is an ionotropic chloride channel. When GABA binds, the channel opens, and negatively charged chloride ions, Cl, rush into the cell. Since chloride is negative, this hyperpolarizes the cell. It pulls the voltage down even further away from the firing threshold.
And this is where benzodiazepines like Valium or Xanax and the older barbiturates work. But they work differently, don't they? The texts make a big point of this.
Yes, and the distinction is literally life and death. Benzodiazepines bind to a separate site on the receptor, and they increase the frequency of the channel opening when GABA is present. They make the body's own GABA work better.
But they need GABA to be there.
They do, which gives them a ceiling effect. Barbiturates, on the other hand, increase the duration of the channel opening. And at high doses, they can actually force the channel open even without GABA. That's why a barbiturate overdose is so easy and so lethal, it can directly shut down the respiratory centers in your brainstem. Benzodiazepines are much, much safer because they are modulators, not direct activators.
Then there is GABA B. What's its role?
GABA B is the slow metabotropic version. It's often located on the presynaptic terminal. When activated, it inhibits calcium channels, which, as we know, prevents the release of neurotransmitters. The drug baclofen triggers this receptor, and it's used as a muscle relaxant for spasticity. It basically tells the motor neurons to shush by reducing their ability to release excitatory signals.
Okay, leaving the heavy-hitting amino acids, let's look at the monoamines. These are the modulators. The texts use the term diffused neuronal systems.
Yes, and we should probably think of this as volume transmission. In that classical synapse we've been talking about, the gap is just nanometers wide. It's like a private whisper between two people. With monoamines like dopamine, serotonin, and norepinephrine, the release sites often lack a tight, dedicated postsynaptic partner.
So what happens to the neurotransmitter?
They release the chemical into the general extracellular space, where it can diffuse over microns and affect many, many neurons in a local area. It's not a whisper, it's a broadcast.
It's like a sprinkler system, bathing a whole region in a chemical.
Exactly. And this architecture allows a very small cluster of neurons, usually down in the brainstem, to tune the sensitivity and firing properties of millions of neurons up in the cortex. They set the tone.
Let's break them down. Acetylcholine, HA.
The cholinergic system is absolutely vital for cognitive processing, learning, and memory. You have the nucleus basalis of Meinert projecting widely to the cortex. And in Alzheimer's disease, these are some of the very first neurons to die.
Which is why we use acetylcholinesterase inhibitors, like Dunpezil.
Right, we try to prop up the failing system by keeping whatever little A sheet is left in the synapse for a bit longer. It's a supportive measure, not a cure.
And the receptors?
You have the fast nicotinic receptors and the slow muscarinic ones. And interestingly, the M1 muscarinic receptor has a very unusual mechanism. It produces a slow excitation by closing a specific type of potassium channel called the M-current channel.
So instead of letting positive ions in, it stops them from leaving.
Exactly. It plugs the leak, which traps positive charge inside and makes the neuron more excitable, more likely to fire in response to other, faster inputs. It raises the baseline excitability.
Fascinating. Okay, dopamine, the one everyone thinks they understand because of pop science.
Dopamine is incredibly complex because its function is all about location. You have the nigrostriatal pathway starting in the substantia nigra, which is crucial for motor control. The death of these neurons is what causes Parkinson's disease.
And then you have the other pathways.
Then you have the mesolimbic pathway, starting in the ventral tegmental area, which is central to reward, motivation, and emotion. Overactivity in this pathway is strongly linked to the positive symptoms of schizophrenia, like hallucinations and delusions.
So an antipsychotic drug that blocks dopamine receptors might help with the psychosis.
But it will inevitably cause movement side effects that look just like Parkinson's disease.
Because you can't block the receptors in one pathway without blocking them in the other. The drug goes everywhere.
That is the fundamental trade-off of our current pharmacology. We lack the anatomical precision.
Let's move to norepinephrine.
Norepinephrine, or NE, originates in a tiny little spot in the brainstem called the locus coeruleus, which means the blue spot. But from that tiny nucleus, it projects to virtually every part of the brain. It regulates arousal, vigilance, attention.
It's the fight-or-flight chemical in the brain.
In a way. It enhances the signal-to-noise ratio in sensory processing. When you're startled by a sound in the night and suddenly you're hyper-aware of every little creak in the house, that's a norepinephrine flood.
And finally, serotonin, or 5-HT, the one with the most complicated family tree.
Oh, by far. Katzone lists over 14 distinct receptor subtypes, and more are being discovered. This diversity is both a challenge and an opportunity. For instance, the 5-HT3 receptor is the only ionotropic one. It's in the gut and the area postrema and is involved in the vomit reflex. That's why drugs like ondansetron, which block it, are such powerful anti-nausea agents.
And all the others are metabotropic.
All the others are metabotropic GPCRs. The 5-HT1A receptor is involved in anxiety and depression. The 5-HT2A receptor is the primary target for psychedelic drugs like LSD, and also a key target for many of the newer atypical antipsychotics. The fact that we have so many subtypes is a blessing for drug designers. It allows us to try and design molecules that hit the mood receptor without hitting the nausea receptor, at least in theory.
Okay, section 6, breaking the rules. We have spent this whole time teaching the classical axon to dendrite forward flow of information, but the brain has a reverse gear.
It does, retrograde signaling. And the most famous and well-understood example is the endocannabinoid system.
This completely flipped the script when it was discovered, didn't it?
Completely. What we found was that the postsynaptic neuron, the receiver of the signal, can synthesize these lipid molecules, like anandamide or 2-AG, on demand when it gets highly active.
So it makes them when it's being heavily stimulated.
Yes, usually in response to a large influx of calcium. And these lipids aren't stored in vesicles. They are made, and then they immediately diffuse out of the postsynaptic cell and travel backwards across the synapse to the presynaptic terminal.
And what do they do there?
They bind to CB1 receptors, which are GPCRs located on the presynaptic axon terminal.
And what does activating the CB1 receptor do?
It acts as a powerful circuit breaker. It inhibits the presynaptic calcium channels, which, as we now know, stops the release of neurotransmitters, whether that's GABA or glutamate. It's a phenomenon called depolarization-induced suppression of inhibition or suppression of excitation.
So the receiving cell is essentially screaming back across the synapse, okay, I'm overwhelmed, stop shouting at me for a minute.
That is exactly what it is.
Yeah.
It is a highly localized, short-term negative feedback loop. And it explains why THC, the active compound in cannabis, which hijacks this system, causes such broad effects on memory, coordination, and perception. It's artificially dampening the synaptic gain control all across the brain.
It's incredible. Okay, section 7, drug action sites. We have built this massive, complex system. Now, how do we hack it? I like to visualize the life cycle of a single neurotransmitter molecule as a journey, with potential traps at every single step.
That's a great way to frame it. Let's walk that path. Step 1 is synthesis. We can walk the factory. This isn't super common, but a drug like metaricine blocks the enzyme tyrosine hydroxylase, which is the rate-limiting step in making dopamine and norepinephrine. No enzyme, no dopa, no dopamine. It shuts down the supply chain.
Okay, step 2, storage. The transmitter has to be loaded into the vesicles to be protected and ready for release.
And we can sabotage that process. The old drug reserpine blocks VMAT, the vesicular monoamine transporter. It prevents dopamine and norepinephrine from getting into the safety of the vesicle. They're left out in the cytoplasm where enzymes like MAO quickly destroy them.
So the neuron runs out of ammunition.
The result is a depleted neuron. It was an early antipsychotic and antihypertensive, but it caused severe depression because it just emptied the brain of all its mood molecules.
Step 3, release.
We already mentioned the toxins like tetanus and botulinum that cleave the snare proteins and stop release. But we can also force release. Amphetamines are a great example. They get taken up into the nerve terminal, and they literally reverse the direction of the dopamine transporter, causing it to pump dopamine out into the synapse, even without an action potential. It's a flood.
Step 4, reuptake. The vacuum cleaner that cleans the synapse.
This is probably the most commercially successful drug target in history. The SSRIs, like Prozac and Lexapro, block the serotonin transporter, SERT. Cocaine blocks the dopamine transporter, DAT. By jamming the vacuum cleaner, you leave the neurotransmitter in the synapse for a longer period of time, amplifying its signal.
Step 5, degradation. The cleanup enzymes inside the cell.
Right. If you can't block reuptake, you can stop the breakdown. The MAO inhibitors, MAOIs, are drugs that stop the enzyme monoamine oxidase from chewing up dopamine and serotonin. Acetylcholinesterase inhibitors do the same for ASHE. It's a double negative. You inhibit the inhibitor to create a positive effect.
And finally, the most direct approach. Step 6, the receptor itself.
This is the direct approach. You can be an agonist, a molecule that mimics the natural transmitter like morphine does at the mu-opioid receptors. Or you can be an antagonist, a molecule that just sits on the receptor and blocks it, like naloxone does at the very same receptors. You are either picking the lock or you are jamming it with superglue.
It's just incredible to think that every psychoactive pill in a pharmacy is just a clever way of manipulating one of these 6 fundamental steps in this very specific cycle.
It is, but, and this is the final crucial thought for our synthesis, we have to remember that the system responds. It doesn't just sit there passively.
This brings us to the real so what of this entire deep dive. We've just spent an hour building a model of a dynamic, integrated, self-regulating system. And then what do? We throw a static, unchanging drug at it every day.
And the system fights back. This is the entire concept of plasticity and tolerance. If you continuously block a receptor with an antagonist for weeks, the cell says, hey, I can't hear anything, and it responds by synthesizing more receptors and putting them on the surface to try and catch a signal. That's upregulation or sensitization.
And the opposite happens with an agonist.
The exact opposite. If you constantly flood a receptor with an agonist for weeks, the cell says it is way too loud in here, and it starts pulling those receptors off the surface and internalizing them. That's downregulation or tolerance.
So the drug stops working as well, or you need a higher and higher dose to get the same effect.
Exactly. Or, even worse, when you suddenly stop the drug, the system is now completely unbalanced in the other direction, which is what causes withdrawal syndromes. We are not just fixing a broken part in a simple car engine. We are pushing on a living, breathing ecosystem that actively pushes back against us.
That really is the core takeaway, isn't it?
Yeah.
Integration, signaling, and adaptation.
That's it. The fundamental takeaway is that CNS pharmacology is the study of how to gently nudge these incredibly complex adaptive networks back toward a state of health without triggering a massive counter-regulatory collapse.
Listeners, your homework is clear. Go to Capsung, Chapter 21, for that beautiful clinical overview. Then go to Goodman and Gilman, Chapter 14, for the molecular deep dive. Specifically, find the diagrams of the G protein cycle and the NMDA receptor structure.
You really need to visualize that magnesium block popping out of the channel. You need to see that G protein splitting apart in your mind's eye. Once you have that mental movie playing, all the drug mechanisms we talked about become obvious.
Thanks for listening to this deep dive. We'll catch you on the next one.